Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is activator and co-activator. So this is related to the transcription. So you just check out different topics related to transcription. I have already explained in my channel. So you just see that, and by understanding that explanation, then you check out this one. Okay. So here this is activator and co-activator. These are the factors, okay, factors that related to the transcription, or you can say that make an influence on the transcription process. Okay. So before going to this topic, I want to say that please if this channel is helpful to you, so you just share this channel with your friends, and obviously subscribe and hit the bell icon for getting the notifications. Clear? So here the activator and the co-activator. This activator is a DNA binding protein that stimulates the transcription initiation. Okay. So this is what? This is one kind of protein. Actually, in the transcription process, different factors that make influence on this transcription process. So these are the only just proteins. So activator is one kind of protein, or you can say that DNA binding protein that stimulates the transcription initiation. And activators, what is the actual role of the activators? These activators either recognize the upstream promoter elements and influence the transcription initiation only at the promoter to which these elements are attached, and other activators target sites within enhancers and influence the transcription of the several genes at once. So what is saying that actually these activators recognize the promoter, okay, promoter elements, and influence the transcription initiation process only. And how this is possible? At the promoter to which this bind or this attached, then other activators target sites within enhancers and influence the transcription. So either this activator may induce the one gene for the transcription, or you can say the many genes, or you can say the several genes at once. That means at once it can make influence on single gene to create the transcription process, or to create the initiation of the transcription process, or it may influence on the different genes or the several genes to create the transcription or to induce the transcription process at once or at a single time. Clear? So its function is that it just recognizes the promoter that is already has bound with the transcript. So here this activator just try to bound with the promoter. First recognize the promoter and then bind with the promoter. Then it enhances the transcription initiation process. Clear? Next is that there are these factors have the two domains. These factors have the two domains, and okay, one thing is that okay, I have to explain another thing is that either these activators attach to the promoter site or within the enhancer. Enhancer is another protein that is also involved in the transcription process. So I will explain about enhancer in another video. So within enhancer it make an influence on the transcription process. So either very simply, if it can recognize the promoter, then it tries to bind with the promoter, or it just within enhancer it tries to bind with the transcript and then the help in the transcription process. Simple word is that. Next thing is that these factors have the two domains. one region of the protein specifically binds DNA. That means this is called the DNA binding domain. So which region? Region that specifically binds with the DNA. This is called the DNA binding domain. And another domain, this is called the activation domain. And this activation domain has important role that activates the transcription by interacting with other components of the transcriptional machinery. So other components means like enhancer maybe is possible, promoter is possible, RNA polymers is possible. So everything is possible. These are all known as the transcriptional machinery. So it activates the transcription by interacting with the other component, not directly activates, just activates the transcription process by interacting with the other components that also involved within this transcription process. Okay, and that is a DNA binding domain. It's directly bind to the DNA. Clear? Now here one thing is that the basic function of the DNA binding domain is to another transcription factor to the proper site of the DNA. So what is saying that? That the basic function is that DNA binding domain that to another transcription factor to the proper site of the DNA. That means it tries to bind with the proper site of the DNA but with another factor that is also involved in the transcription process. Clear? And the activation domain that independently stimulates the transcription process by interacting with the other proteins. So its role is another thing that independently stimulates the transcription process. Okay. So one thing is that the basic function is that the first one, that is the activation domain. Its function is that it can bind with the proper site of the DNA but with the other factors, and activation domain that independently stimulates the transcription by binding to the other factors. So two main difference is that the DNA binding domain that attach with the DNA or bind with the DNA, then the another factors also bind with that. But here in the activation domain that independently stimulates the transcription process, directly bind with what? By the other factors, not directly with the DNA. Clear? And there are some common type of the activation domains these are known as the acidic domains. And example you can say these are the acidic domains, some glutamine rich domains and the proline rich domains. These are all involved within the activation domain. The gene regulatory proteins that do not themselves bind DNA but assemble on DNA activator proteins are termed as the co-activators. Now the term co-activators has come. What is this co-activators? The gene regulatory proteins that do not themselves bind DNA. That means they cannot bind directly with the DNA but assemble or try to assemble on DNA activator. Okay, activator proteins, and these are termed as the co-activators. So these are not directly involved in the transcription process by binding with the DNA. They attach with the activators. That's why their name is the co-activators. Clear? Now, these co-activators, two main classes these co-activators have. One is the chromatin modification complexes, another is the chromatin remodeling complexes. So these enhances the accessibility of the template DNA to general transcription factors or the specific activators. Okay, this is not general, I think this is generic. Okay, one thing is that I have to check first one. Okay, no, this is the general. The word is the general. So main important thing is that these two factors generally enhances the accessibility of the template DNA for what? For the transcription process. And obviously this try to due to the presence of these co-activators, there are the chances of binding of the transcription factors or the specific activators with the DNA. Clear? That means co-activators has played, or the main important role played by the co-activators is that it create an influence by which these activators and the transcription factors, they can bind with the DNA. Clear? There are some important example, that chromatin modification complex, this is the HAT, and CBP. The full form is that histone acetyl transferase, and this also acetylates histones, and CBP is that crane binding protein. This is the HAT activity. And another important example of these chromatin remodeling complexes, this is the ASWI or SNF, and this is the So ISW1, this is the imitation SWI2, SWR1, and one common example that is present in the eukaryotes as known as the eukaryotic activator. This is the GAL4, okay, GAL4, and it activates the transcription of the GAL1 gene in the yeast. What is the role of this GAL4? Its role is actually it helps in the transcription of the GAL1 gene in yeast, and GAL1 gene that codes for the enzyme and convert the galactose to the glucose. So what is the function of GAL1 gene? It helps to convert the galactose to glucose. So this is very brief topic, or you can say that is very brief explanation of the activators and the co-activators that are involved in the transcription initiation process. So another important thing is that enhancer also. You just here saw the term. This is the enhancer. Where is that? The enhancer. This enhancer also important factor that plays this is important factors that also involved in the transcription process. So I will try to explain about this enhancer in my next video. Okay, so you just continue with my channel, and thank you.